All right, good afternoon. We are going to start Chapter 12. Chapter 12 has 4 parts, and we'll see, this is part 1 of the 4 parts. It's how signaling starts. It's the basic principles of signaling. And Chapter 12's whole topic is actually related to Chapter 11. We talked about membranes in Chapter 11. In Chapter 12, we talk about, okay, the cell has this great membrane, which is supposed to be really a barrier between the outside world. But of course, the cell needs to respond to the outside world. How does the cell do that? And so we're going to talk about how a signal from outside the cell causes a change inside the cell. So let's narrow it down. Let's show the, here's a video of where we start an immune response. So we start to we basically shoot the lymph node of a mouse with a laser, and then we see what happens. This is damage. The immune system should be coming to see it. There's the damage, and the immune cells are shown in red and green. And you can see that the immune cells are sensing something from outside the cell, and they are moving to come to the point of damage to do their immune work, to try to treat the damage and or stop the thing that is damaging the cell. These are neutrophils, by the way, if you've had the immunology course, that we'll talk about that. Here's another example. You have 2 cells on pipettes, and you can move them around. And so you have a cell, there's an immune cell on the pipette that's a neutrophil, and it's trying to go toward, it's got a cell that is its prey is on the left. And the little cell is secreting some kind of signal that the neutrophil is detecting, and it keeps trying to go for it. But you see how they move it around, and then the neutrophil finally gets its lunch, and it eats the cell right there. The neutrophil is responding, and what's inside the cell is changing its shape to match what's outside. And it changes its shape to push the neutrophil toward wherever the prey cell is. This is exactly like a predator-prey relationship. But this is a predator-prey relationship that is on a cellular level, which means that we can break it down. There must be some kind of receptor on that neutrophil that is detecting some kind of molecule from the other thing. It's not detecting photons. It's not detecting, some kind of, it might be detecting magnetic fields, but let me tell you it's not. It's some kind of chemistry that's going on that is being detected. How does the chemistry from outside the cell get through this barrier? We just spent this whole chapter talking about how great membranes are at keeping things on one side or the other. Well, here we want something to cross through the membrane. How do we do it? Here's another example. This is a neutrophil that is chasing a little tiny bacterium. It's a little speck. But you see that no matter where the bacterium goes, the bacterium is like changing direction. It's not doing it on purpose. It's just sort of doing it randomly. But the neutrophil is literally chasing it down because it has receptors that detect some molecule, and there it is. It's just caught the bacterium. So it's pretty amazing that you can have these cellular things that sense their environment and change their direction in order to chase something down. They are chasing something, and this tells you something they're chasing. Here's a slime mold chasing the pipette, and what's in the pipette is a small molecule called CAMP. And so you can see how when the CAMP is moved around, the slime mold will move towards it, and then you move it, and the slime mold's like, oh, wait, where'd it go? And it goes up to the other way. Wherever you move the pipette, clearly the slime mold is sensing the CAMP, and it's moving down the diffusion gradient to go toward greater and greater concentrations of CAMP. Because it's a slime mold, it doesn't get bored. And there's another one coming in and saying, oh, is that some CAMP for me? And there's a third one coming in the bottom there. So this is a really great example of how a simple chemical is attracting the slime mold. So we can get it down to these. These molecules, there's more information about these at this video. I think these are really great examples of how you can chase down not just a bacterium, but a single chemical, the CAMP. And then you can do biochemical experiments that CAMP is somehow fitting into a receptor on the cell surface. And that is called the first messenger. The first messenger is the stimulus that reaches the cell, that binds a receptor on the cell and tells the cell to do something else. So in the case of CAMP, you remember that we talked about what the structure is. So just to remind you, it's fair game to ask you what the structure is, but here's the CAMP structure. And there could be other receptors for CGMP. Maybe it cares whether it's got a GMP or an AMP, or it could have a receptor that binds to both. The main thing is, starting with an experiment like what we showed you, we can now do biochemistry to ask, what is the receptor? How specific is the receptor? Does it care whether it's GMP or AMP? And what happens inside the cell after the event outside the cell? By the way, it's showing receptor here because that's the common thing. The other option is there could be a transporter that responds and opens to let the AMP through, and then the AMP itself could be causing the signal inside the cell. And basically we just got to look at it to see what kind of protein is in the membrane that is causing a change inside the cell. Inside the cell we have what's called a second messenger that's produced inside in response to the first messenger. Now, usually the first messenger is some kind of complex signal, a peptide, a protein, a lipid. In this case, it was kind of weird that it's CAMP for the first messenger, because usually CAMP is a second messenger produced inside the cell. Commonly, it's easy to take a nucleotide. You've got lots of ATP floating around inside the cell. And so what the receptor can do inside the cell is it can grab an ATP, change it, refold it into a CAMP, and then send that out from it through the rest of the cell to alert the rest of the cell to the fact, hey, there's a bacterium over here. Let's change our shape and let's move in that direction. So that's one thing not to be confused about. CAMP is most commonly a second messenger. In the case I showed you, it was a first messenger for the slime mold. And in evolution, probably it was changed in evolution at some point. But the main thing is us humans use CAMP as a second messenger inside the cell to produce signals. Here are the kinds of first messengers you can have. Light itself is a first messenger that changes a receptor inside your eyes. Antigens are first messengers. Growth factors, odorants, tastants. So we're going to talk about at least 3 of the 5 senses, because sight, taste, and smell all start with first messengers from outside the cell, causing a nerve to fire inside the cell. So here are some bacterial second messengers, and you can see CAMP definitely made the list. cGMP can make the list, and sometimes bacteria make more complex second messengers if they need to send a different kind of signal. And this is the example of some of the complicated, but notice that these are usually just refolded and reorganized nucleotides. You make a specific shape of a nucleotide, you send that out, and the rest of the cell has proteins that are looking for that. When it binds those proteins in the cell, it causes another change in another part of the cell. And it's useful to have small second messengers because you want to make a lot of them and then flow them out, diffuse them to other parts of the cell. Here's just one of my books. This scientist is one of the most interesting scientists that I've read, and he talks about how in this book about order out of chaos, it's like, how do biological systems work? How do we have something that works from the chaos of chemistry to result in biology? And for example, he uses slime molds as an example. So it's the exact example that I just showed you. And he talks about how CAMP, being both a first messenger and a second messenger, could explain how signaling messages were originally evolved in the first place in something like a slime mold. So the fact that the slime mold was responding to CAMP as a first messenger, but also many organisms use CAMP as a second messenger, that could be a hint to how these things evolved and how they result in stable oscillations, things that are changing but also returning to their original state, which is the basis of biology. That's kind of philosophical, but I just want to mention that that's a clue. When we see some of these things, CAMP can be a first messenger, but more commonly it's a second messenger. Maybe that's a clue to how evolution worked. And I think that's fascinating. But the main thing that we need to talk about here is how does signaling work? And there's 5 main principles in this first part that you want to be aware of. The first one is specificity. It means a particular molecule will bind a particular receptor. In the case of the slime mold, CAMP would work. If cGMP didn't work, we would say that it was specific for CAMP. It's just a question of trying things to see what works and what doesn't. And that means there's a molecular specificity. There is a specific chemical interaction onto the protein. The second thing that happens is amplification. You only have one or a few first messengers, and yet inside the cell you can see a flood of second messengers and a flood of proteins, much more changing inside the cell than starts the stimulus. That means that usually what you see is you want signaling to amplify the signal. You want to be able to respond to a small signal and then to take it and have multiplying results from it. A small signal can have a big change. The other thing that you see in signaling a lot of times is modularity. You have interchangeable parts. So something you learn from slime molds actually may have a homologous component in higher organisms or even in humans, and that turns out to be the case. It looks like evolution has taken a signaling system and tweaked it when things evolve to result in different results in different animals, but the same principles underlying all of those. That's really good because it means we can understand signaling and we can do experiments on yeasts and get conclusions for how signaling works in human cancer. The other thing that happens with signaling is you want it to be able to be turned off. When you have it be turned on by a specific interaction, well, that's great. But eventually you need it to be desensitized or adapted. Imagine what would happen if your eyes would never adapt to bright sunshine. When you walk out and the sun would be out there, it would dazzle your eyes. But what happens is that the dazzling signal eventually gets turned down because you don't need a signal that's always on, might as well not be a signal at all. So you need to have some kind of way to turn down the signal, to desensitize or to adapt to the new configuration. And finally, signaling can have not just one signal can affect things. Sometimes you need to add together multiple signals. So you need to integrate them. Signaling should have some kind of integration where you can have two signals that can add together, or maybe two signals can cancel. If you have this signal going on, but this signal is also going on, the other signal can turn off the first one. These are all related, and these are 5 aspects that we want to go through. So here's little pictures about each of them. Specificity is shown in a picture by having a receptor binding its ligand. It's a lot like enzyme kinetics. You have a complementary site, and you have substrate 1 that will fit into the receptor. Substrate 2 is a different shape, and it won't work. It's specific for substrate 1. And so this has a chemical reflection. The shape of the receptor should match the shape of the substrate. This is our second one, and if you see there is an amplifier on the left side of the screen, a Fender amp, and the little twang of the string on the electric guitar is amplified into a big loud sound by the amplifier. And that's the same way in which you have a little twang of an initial signal, a photon or a CAMP molecule or something like that. And the thing that you want signaling to do is to be able to take that and multiply it. And one way that signals do this is they have a cascade of enzymes. Basically, the signal activates enzyme 1. The thing you know about enzymes is they don't have to produce just one substrate. In fact, they usually can produce tens or dozens or hundreds of substrates. If enzyme 1 is turned on, it can produce 3 substrates that react with enzyme 2. Enzyme 2 will convert those 3 substrates and it will activate 3 more enzymes. So enzyme 1 activates 3 more enzyme 2s. Each enzyme 2 activates 3 enzyme 3s. And you can see from this, you can have one signal that results in 9 activated enzymes from just this simple example. So this is called an enzyme cascade because it's spreading out like a waterfall. It's falling down and spreading out. And so they call it an enzyme cascade. The concept of modularity comes from the fact that you have different levels, and it comes from this level of amplification. If you have 3 different enzymes and they are arranged in an enzyme cascade, they can also be modular. Like you can take one enzyme and you can evolve it to work with a different thing. So we say that proteins form multivalent affinities. They have different levels, and you can have a signal that comes through to a response, and then you can evolve one part of that in one cell to change the signal response mechanism, while the other cell keeps the same thing. And you will actually see that the thing that modularity usually works on is it works on phosphorylation. You see phosphorylation over and over again in all sorts of contexts. Different proteins will phosphorylate the next one. In fact, that's what's shown in this amplification. The idea is that if the green triangle is a phosphate group, the signal causes a phosphate group to be put on enzyme 1. You have 3 more phosphate groups put on enzyme 2. Then you have 9 on enzyme 3. Amplification and modularity go together. The idea of phosphorylation being a signaling molecule is itself an idea of modularity, because you can take that signaling cascade and you can evolve it to be slightly different in another type of cell. It's still dependent on phosphorylation. What's different is the proteins. And so the phosphorylation, in a sense, is an interchangeable part. There's a modular thing. Now, this is the thing I want you to pay attention to. Hopefully this is on a fairly big screen. I think it will work on your phone too. But I want you to stare at this point right here and just stare at it. And it works for me, at least looking on this. If you keep your eyes perfectly still, you'll start to notice something. The outer rings will start to disappear. And in fact, the middle ring won't appear to change, the middle dot. But stare at the dot, and the outer rings should slowly disappear, because what's happening is they're being desensitized. Your eyes are getting the photons, but your attention is focused on the center. And so your eyes are saying, I'm getting a signal from the outside, but you don't seem to care about it. You just care about that dot in the middle. So I am going to turn off the exterior signals that appear to be less important to me, because you don't need them anymore. These kind of optical illusions are examples of desensitization or adaptation. And there's some kind of feedback circuit that after a certain amount of time of sending the signal, the receptor turns itself off. Here's what I want you to do. Stare at the cross. What do you see happening? I basically see what looks like a green dot erasing the pink dots. But look away from it, like blink, and you'll see that no, there's no green, and the pink dots are always there. This is another example of color receptors desensitizing. And the one thing that's interesting is you notice that part of the desensitization is that the pink dots become green. At least in my view, it's sort of a blue-green. I mean, your color might be slightly different. But yeah, just look at that, and to me this is amazing. And what breaks the spell is when you move your eyes. Hopefully this is working for you. If it's not working on your phone or something, try looking at it on a computer monitor, because it's working for me on a computer monitor right now, full screen, bigger the better. Here's another thing where you look at these spheres and you see the colors on them. You know, what color are these spheres? And actually now I'm too close to this, because the spheres look like mostly the same color to me, but they're exactly the same color. And if you lean back, that actually looks like they're spheres that are red, blue, and green. But when you zoom in, you can see that they are all the same color. So this is another thing, and this is a case where the colors are sort of interacting with the beige parts of the sphere to make the beige parts appear colored. Yeah, I can see a little bit of it. It's not that distinct when it's too big, but you can still sort of see it. And the farther away you get, this one works better. So finally, when you have the signal, you have two signals that will integrate. And so you can have the idea that you have two different signals, and receptor 1 is activated, receptor 2 is activated. But what if receptor 1 causes CAMP to be made, and receptor 2 causes CAMP to be degraded? Then you have a negative integration, where you have signal 1 and signal 2 both around. The CAMP gets broken down as soon as it's made, and you end up with no signal. So the thing is, if you have signal 1 and signal 2 related in this way, you have an integration. Just notice it can happen with the increase and decrease of molecule X, or it can happen with the increase and decrease of ions across the membrane, which would be membrane voltages. Remember, we talked about how membrane voltages can be affected by moving ions across. And so this is one way. This can be a concentration, or it can be a voltage, which is a general concentration of charged molecules. This is going to be important. So the power of integration is actually what most brains are doing when you're doing an optical illusion. Like right here, if you look at this, it looks like these are a spiral to me. But when you zoom in, it's not really a spiral. It's actually just a circle of squares. And in fact, if you measure it, it's perfectly circular. but your brain is integrating the shapes and it's confusing itself. And so there's a circle. It's not a spiral, it's a circle. Here's one last illusion. Stare at the dot. Okay, and I'm going to do it. And right there, I can see colors. But blink, and the colors go away. It's actually gray. There's no color there. The only color is the after image of the colors that you start with. Do it again. So stare at the dot. It's blue. And what do you see? I see yellow on the left and I see red on the right. But that's not really there. When my eyes move, all of a sudden I see the reality that it's black and white. That's another example, and this is one of my favorite illusions. I could watch this all day. So the main thing is, these all work out. What's happening in the slime mold that we showed you? The CAMP is binding a 7-helix transmembrane receptor that we call a serpentine receptor because it's all in one chain, and so the chain is going back and forth like a serpent across the membrane. And it looks like the slime mold does this because of the modularity of the response. We actually use receptors a lot like that to detect all sorts of different first messengers ourselves. And so you see you have the CAMP outside the cell. It never actually crosses inside the cell, but it causes a conformational change to happen inside the cell. Somehow that is moved through the shape changes of the 7 helices, and the signal is sent inside the cell. A cascade is begun. And the slime mold gets the signal, move in this direction. And it does all the machinery to move in that direction. So there's 4 basic types of plasma membrane receptors that we're going to talk about. The first is actually the type shown on the last slide. They're serpentine receptors, but they're called G protein coupled receptors for reasons we'll explain in a second. There's an external ligand that binds to a receptor, and it causes that green G protein to be activated, move to another enzyme, and to produce a second messenger. We're going to talk about all 4 of these. Then there's receptor tyrosine kinases, where you have a ligand coming in, and then what you have is you don't have a G protein, but you just have phosphorylation started inside the cell. Phosphorylation is called kinase activity, and so that is what the kinase is directly on the receptor in that case. Then, instead of having phosphorylation be your second messenger, you can have a nucleotide be your second messenger. And so, for example, you can have a receptor that's a guanylyl cyclase. That means that when it's activated, instead of having a kinase cascade on the inside, you'll have a direct making of a second messenger. So you'll directly make, like cGMP in this case, from the GTP that's around. And finally, the 4th kind is where you actually have a transporter or an ion channel instead of a first messenger. The second messenger is sort of voltage. What you have is you have a ligand coming in, and you have the receptor open up, and ions move across the channel, changing the voltage, and this will send a signal. This is how the senses work. So we'll talk about these 4 different types as we go through this lecture. Notice that the second messenger is being produced by the enzyme that interacts with the G protein here. The second messengers are produced more directly for the other kinds of receptors that we talk about. And just notice particularly the gated ion channels that builds directly on section 11, chapter 11 that we just finished talking about. All right, and that is part 1.